gap in the cytosol, how does it, like, what role does it play in it releasing the proteins inside the lumen of the nucleus?
What role does it play in RAM being inside the nucleus?
No, so you know how we activated we attached GTP and when it's in the nucleus, and so it goes up in the membrane. Yeah, through the gap. Yes. Then in cytosol, it's like it's the GTP turned to GDP by the gap. In terms of it releasing the actual NLS-bound proteins inside the nucleus.
So yeah, so let me get to that. So you're getting to the next step.
Okay.
All right. So just hang on that thought for a second. So the net effect of this is we have a different activity of RAN in one side or the other of the nuclear membrane. All right. So from this picture, you should be able to conclude things about, for example, RAN gap. Why is RAN gap not in the nucleus? Two important factors in there. So who can give me one factor?
It doesn't have an LS.
It doesn't have an NLS. How about a second factor somewhere else?
Is it too large to?
And it's too big to fit. Right. So if it was small, it would have gone through the hole. It's not. It's large, and because it's large, cannot enter the hole, and it doesn't have a signal to get into the nucleus, so it's not transported to the nucleus. All right? What about GEF? GEF is also, you wouldn't know, but GEF is also a large protein, and GEF has itself a nuclear localization signal, so it is transported into the nucleus. Another little feature that RAN-GEF has is illustrated here, which is that it has the ability of binding to chromatin. So it not only goes into the nucleus, but then is firmly anchored inside the nucleus by binding to chromatin. That might seem like a trivial point right now, but when we come back and talk about mitosis, that will be a very important thing to remember, so just store that in a special part of your brain. All right, so we have all components now. We have proteins with signals, we have transport receptors, import or export, and we have RAN, right? So now we have to put all three together to explain how we transport proteins from one side to the other. Outside to in to out, right? So there's a variety of diagrams. This is, oh yes, hold on. This is just a little illustration that I made about RAN activity. So now I have to play it because I've made it. So inside the nucleus we have lots of RAN GTP, and outside the nucleus we have lots of RAN GDP. All right. So what does active RAN actually do? So of course one of the things it does is binds GTP and then hydrolyzes GTP. Right. But besides that, once it has RAN, once it has GTP bound on it, what can it do? So it does these four things. It can bind, import, and export receptors. If it has GDP, cannot bind to either of them. And it will, we're going to go through all of this in the next few slides. It blocks the ability of import receptors to bind to anything else, to their cargo proteins. But it does the opposite to the export receptors. That difference is going to be key in making the thing work one way or the other. And finally, inactive RAN with GDP bound in it is inactive, doesn't interact with anybody other than with RAN-GEF to pick up a new GDP. All right? So that's illustrated here in this figure from your textbook. That I find a little bit confusing. That's why I wrote it all in other ways. So here we have an import receptor. This little handle there is there to illustrate the negative amino acids on the receptor that interact with the red positive amino acids of the cargo protein. So that red part there illustrates that nuclear localization signal. Please remember that nuclear localization signal is part of the polypeptide of that protein. It's not something attached to it. That signal is part of you. It's like your hand. Right? Your hand is not attached to you, it's part of you. So the nuclear localization signal is part of the protein, because it here looks attached to it. So the import receptor can bind to that signal right there. But this interaction can only happen if RAN doesn't get in the way. If RAN has GTP and is all pumped up, what it will do is will bind strongly to the import receptor. It will prevent it from binding to the cargo protein with the NLS. So here you have an either or. Either you bind the cargo protein or you bind RAN with GTP, but you cannot bind both for the nuclear import receptor. All right? So this is what's going to control loading and unloading. Binding on one side, releasing in the other. So same thing is true for export. I'll come back to that. Your textbook has this complicated diagram. It's very accurate. You can go there and read it. But I'm going to use an animation here to illustrate this point. Right. So let's just consider import of proteins from the cytoplasm into the nucleus. So we have to have a protein with an NLS. So this might be a transcription factor. This might be DNA polymerase, RNA polymerase. This might be all the proteins that are used to form ribosomes. They also have to be synthesized in the nucleus, in the cytoplasm, transported to the nucleus. There's so much protein that has to go into the nucleus all the time. So they have to be able to interact in the cytoplasm, and they do. They do because there's no active RAN on the outside to get in their way. The binding is strong. They can bind in the absence of RAN GTP. Why is there no RAN GTP on the outside?
There's the RAM gap on that side.
Because there's RAN gap on the outside that has promoted it to hydrolyze its GDP. Now it's all outside, is only in the GDP state, which will not block this interaction. Cool? So they will be able to diffuse, now this is the key word, diffuse across that nuclear pore complex with the jelly stuff in the middle. That is a process of diffusion, not of active transport. We're not using the energy of ATP to move across from one side to the other, just simply by random diffusion. Because it's random diffusion, you could potentially even say, okay, maybe this pair just went halfway through the nuclear pore, and then they just turned around and went back out. That's the way diffusion works, all right? Now, statistically speaking, you're going to end up moving the other way, and as soon as you touch the other side, in the nuclear side, what protein are we going to encounter? RAN-GEF. And RAN-GEF has activated RAN-GTP, so there's lots of active RAN-GTP inside the nucleus, which will be able to bind to the import receptor. In doing so, boom, it will release the cargo. So we have achieved the goal here. We have, our goal was to bind a protein on the cytosolic side and release it in the nuclear side. When you are writing exam questions about this, be very sure to use that language. Cytosolic side, nuclear side. If you use inside and outside, very commonly you will make the mistake of saying outside the cell. It's going to be a natural mistake, and that's going to be not correct. So it's easier if you get used to just saying which side, nuclear side, cytosolic side. All right? Clear? Questions? Yeah.
The RAN GTP binds in a different location and how does it change?
Yep. That's was tried to illustrate in the other textbook figure. It binds on one side, and when it's bound on that side, then it cannot bind the protein with the NLS. So are we done? Rhetorical question, obviously the answer is no. Why not? Because we need to return that in. At this point, this import receptor is captured, is not able to do anything else. We're not going to dispose of it. We need to liberate it to do more rounds of import. So how do we do that? Well, that import receptor, does it have the ability to go out by itself? Yes, because it can, remember, it has the ability to interact with the hydrogel and move through the hydrogel by diffusion. And this time it's going to drag a RAN GTP to the outside with it. So maybe through the same pore, maybe through a different neighboring pore, doesn't matter. It will recycle back to the cytoplasm. As soon as the pair reach the cytosolic site, they will find RAN gap out there. And that means RAN will hydrolyze the GTP. And then that will cause it to release the receptor. So now the receptor is free in the cytosol, once again, to do another cycle of import. So you don't need that many receptors, transfer receptors, to be doing continuous transport of proteins into the nucleus. Same thing is going to be happening with export receptors. Export receptors are going to be kicking things out, and then they need to return. So you're going to have this one in, out, and the other one the other way, yes. And all going through the same pores. The pores are just a piece of jelly. There's no directionality to it. So through the same pore, you have the traffic of lots and lots of proteins in, lots and lots of RNAs and proteins out. Let me ask a question from my local expert. Do you know how many proteins traffic per minute or second from one side to the other?
Yeah, I don't know. So in yeast, there are about 300 nuclear pore complexes, and they're making about 2,000 ribosomes a minute. That takes about 200,000 proteins coming in per minute, and that's just protein transport. So hundreds of proteins, thousands of proteins per minute.
Okay, so gobs and gobs and gobs. All right?
That's the same
Is that
time-
accurate enough? Gobs? Thank you. All right, so just tons of traffic through these little holes, all right? All right, so that's import. So let's go through export. Oh, yes, question.
So, what happens to the RAN GDP once it's back?
Oh, great question. So we've kicked RAN out, right? Let me save it for a minute. Let's do export, and then we'll come back to that, all right? Because it's going to aggravate the problem. So let's do export. Export, we're going to start inside the nucleus. We need to grab a protein, a cargo protein, we need to grab it and put it outside. And we need to grab it because it's big. If it was small, it would just go out by itself through the hole. But it's big, so it has to have a special signal, a nuclear export signal, that it needs to be recognized by a nuclear export receptor. But this export receptor will not do that unless it has the help of RAN with GTP. So you need the three coming together into a complex. All right? And this complex will only form because of the help of RAN GTP. The export receptor is unable to bind unless it has bound to RAN GTP. So it's the opposite of the import receptor.